Hello, everyone. I hope you all had time to catch something to eat or have a nice break. So I will be I will be presenting module 4.1. This was supposed to be the first module of today's date, but we ran over a little bit, but we will continue building up on what James was presenting. The module is going to be building upon what everyone on what James was discussing earlier. But specifically in the context of how to model intracellular processes, although we will talk about how could this workflow could also be extended or used to represent processes that are happening at bigger than tissue scales, especially at systemic level processes. So again, this is the first module of day 4, and I'm suspecting this model might run over a little bit. I have a couple of interesting examples I would like us to walk through thoroughly. If we have a little bit of time after this presentation, then TJ will be presenting the principles of how to use Boolean networks in CompuCell. In the first part of the module, I will be talking about how we can use CompuCell to do multi-scale models. And by multi-scale models, we mean models that incorporate or integrate processes happening at widely different biological scales in terms of time and spatial scales. And we will learn how we can, once we've specified such models using antimony strings, how we can load these models into CompuCell, and then how we can load them into cells, depending on what the problem of interest is. Then I will be discussing 2 examples. I will so Taru, no, because of 4.1.5 is just an example that we're gonna be using a repository on the internet, and that's me just walking you through how to look for those repositories. Then I'm gonna be discussing the delta notch and the cell cycle examples, and we'll go into the details of that later. So far, we've focused on the capabilities of CompuCell of representing processes that are simulating processes that occur at the tissue level. So these mostly pertain the processes that involve the mechanical properties of cells, such as their where are they located, how much of the space they occupy, what shape they have, whether they move or not, whether they stick to each other, and whether and processes that we have roughly modeled just from this tissue level. We have modeled very basically, such as how cells can differentiate, how can they divide, how can they die, if they polarize or not. And we've also presented at the tissue level what the interaction between these specially resolved or specially distributed cells have with surrounding chemical fields in terms of how this might regulate cellular behavior. We saw that with controlling cell growth based on the amount of resources that the cell has available to them, and how the cells can secrete and uptake from their environment. However, the processes, biological processes, and the problems that also as biologists or physicists or computational biologists are interested, often span over multiple scales, biological scales. Some of these processes, so again, so the reason the screen is small right now is because I'm planning on using Tellurium. I'm sorry, I'm planning on using CompuCell3D NanoHub, so I have to have my my screen split, and I was just showing this part. But again, the presentation is available in the Google Drive in case people want to have them as a reference side by side so they can look at the details more closely. But as I was mentioning, the biological, often the biological problems involve processes that span over different scales, processes that have occurred at the whole organism level, that interact with tissue level processes, that in turn interact with processes happening at the intracellular scales or subcellular scales. And CompuCell has the capability of integrating these processes as we can model the processes and incorporate them into our simulations, depending, and how we incorporate them, it's gonna of course depend on the particularities of the problem of interest. And this in this particular image here, this was a this is taken from a paper from our group. Sluka, it was the first author on this paper, and they were interested in looking at acetaminophen metabolism and the interactions between whole body PBPK models with models of the flow in the liver, and then interactions and the metabolism of the chemical compounds or the active compounds inside each cell. Julio, this is another example of a model developed by people in our group or related to our group. This one in particular was developed by Julio, and it's a multi-scale model of somatogenesis. And it does also integrate multiple scales, specifically scales where something is being introduced into the cellular domain, a particular signal, and this signal then downstream regulates the differentiation and polarization of cells. When we think about the different types of subcellular modeling that could be of interest from the perspective or that could be integrated in CompuCell, we have many options. And in principle, we are able to integrate whatever model we want of subcellular processes with our CompuCell simulation. And the important thing, right, what is going to turn our model into a multi-scale model is our ability to tie some of the properties of our CompuCell simulation to some of the outputs of the subcellular models. So for example, we could develop a model of a cell cycle where, let's say and we will see this later on, the state of a variable in that intracellular model determines or triggers cell division. And there is many ways in which we could implement this subcellular model. In this particular module, we will focus on modules to use on processes model using reaction kinetic models. These are the types of models that are antimony string support. But later on, TJ will be showing how other types of models, such as Boolean networks, could also be used to represent the same subcellular processes. Another interesting example that we will see later on is that delta notch. James already mentioned, talked a little bit about the notch signaling pathway. We will see how we can, using a simple set of differential equations to describe the state of the signals inside each cell, produce a model, a simulation that generates a characteristic patterning of delta node activation. So in terms of how to or where we integrate these reaction kinetic models, so there's multiple options available to us. And again, what options we decide or go with are going to depend on the particularities of the problem that we are interested in addressing. So for example, we can have a model, a subcellular model that describes processes that occurs in all of our cells. And in that case, we can have that model and we can load it in all of our cells, and then we can also particularize it if we want it. An example, for example, if we have a model in which all of our cells are capable of dividing, then we might incorporate a cell cycle model into all of our cells. If we have a model of viral replication and all of our cells are capable of taking up and reproducing virus, then we will load this viral replication model into all of our cells. We can also load the models by cell as a particular cell ID, let's say, because there is a special cell in our simulation that we want that cell to have a particular subcellular model. We could do that as well. We could model it, we can incorporate it by cell type. So if there is just one type of cell that can, that in which the process of interest occurs, we can just load it into the cell. So for example, we saw an example where we had a stem cell and that was the only cell capable of differentiating. So a model of stem cell differentiation, we could load it into all of the stem cells in our simulation, and then all the terminally differentiated cells would not have that model. And finally, we can load the model as what we call a free-floating model. And in this part, this is a special case. And we usually interpret these free-floating models as processes that affect the whole simulation domain more or less uniformly, and that describe processes happening at higher biological scales. So for example, if a compound is being transported, we interpret something being introduced into our simulation as a compound being transported from the blood to the tissue, then we can simulate it that way. For example, if we had a model where the characteristics of our, let's say we have a infected tissue patch, this is something TJ described already, and we use information from that tissue patch to recruit immune cells in the lymph node, then we would use a free-floating model as a way to describing how information is transported or transmitted from the local site of infection to a whole organism level. There are multiple ways in which these reaction kinetic models can be specified and simulated, and how they can actually be integrated into CompuCell. We're gonna focus on the antimony specification that James discussed earlier, and we can then simulate these models in CompuCell directly. But there's other options as well. You can there is specialized software that can be used to develop such reaction kinetic models, and they could be translated into SBML files. And then these SBML files, which is just a markup language, use a standard markup language used to specify reaction kinetic models, can also be imported into CompuCell. And my last example does just that. So this is a lot in this slide, but I just wanted to give a quick overview, and we're gonna go into the details of all of this, but the syntax and the reference to antimony models is always just gonna look similar to this. We're gonna define our antimony model as a string. Then we will import and load our antimony model. Either depending on the specific option that we're interested in, and we will explore some of those options. And then we always have to time step or call a special function to ensure that this that the CompuCell integrates our antimony model. So this time step underscore SBML is always gonna be it's gonna be required in all of our in all of the simulations that involve an antimony model or any type of SBML model. And there are ways for us, as I was mentioned earlier, the way we build this multi-scale models is by connecting the outputs of subcellular models with properties of our CompuCell3D simulation. And the way we do that is by retrieving or reading and writing the variables of these models. And then there is specific syntax that allows us to both read and write from the models that we have loaded into the cell. So the way I like to think about it is I imagine out my cell, and my cell has inside a model, and then I can read the outputs of that model inside my cell and do whatever I need to do with that read off at the tissue level. And then if there is a downstream effect from the tissue level into the intracellular processes, then I write on the same model output from the SBML model. And we will see, this is conceptual at this point. I just wanted to introduce the ideas, but we will see examples of all of these options in the next couple of exercises. So now let's go ahead and have our first exercise. So in this particular example, what we're gonna do is we're gonna just generate a genetic simulation template with two cell types, proliferating and quiescent. We're gonna specify their contact and volume constraints. I'm not sure we're gonna use the volume constraint. We're gonna specify both, but we're gonna, the contact and volume constraint. And then we're gonna define a basic antimony model. And then we're gonna load the model as free floating and make sure that CompuCell can step it. And then we're gonna retrieve the data from these free floating models such that we can plot them. And then, I'm sorry. After 1,000 Monte Carlo steps, then we're gonna change the value of manually, we're gonna change the value of our antimony model. I'm sorry, 1 second, I couldn't see my cursor. So the first thing is we're gonna generate the template simulation. We're gonna generate a new project. Again, this example is already loaded in the demos directory, but we're just gonna build it from scratch because we have time. So we're gonna generate our first exercise. No space. Always forget to change the directory I can save stuff into. Okay. We're just gonna leave it like this because for the purpose of this exercise, we don't care that much about our simulation domain. We're just gonna add two cell types, proliferating and quiescent. We're not gonna have any chemical fields. And I mentioned that we want the contact constraint and the volume constraint. Yes, absolutely. So far I've just created a simulation. I didn't change any of the dimensions of any of the general properties of my simulation. I just created two cell types. Yeah, you can, I mean, the neighbor order, you can always, again, I keep forgetting that you can always prefer setting it to 3 just to avoid any square lattice effects. But really, for the purpose of this particular module, we're not going to be using the cells. We just wanted them to show that there's no interaction between cells and a model. And then we've selected contact and volume as the constraints we're going to use in our cells. Okay. Then I open my simulation, and then the next part the next part I'm gonna write a basic antimony model. And to write my antimony model, so I did wanted to say that this exercise will basically generate something similar to the template code that you were using in the previous exercise when you were writing your antimony model and plotting them to using CompuCell. So that's basically what we're gonna build together in this exercise. So the first part, we're just gonna define an antimony model. Let's call it model string. And it's always a multi-line string. And then I always like to type when I'm writing my equations, my parameters, and my initial conditions. So if I have to come back and read my code my model later, I know what part was intended to represent what. And this is a very simple model. In this model, A goes to B. We can think of a particle becoming B, and then a particle A becoming B, and then the particle B becoming A. So it's a bidirectional reaction. I always call them J's for the reaction rates. And that one is A goes to B at a rate that is K1 times A. And the next equation is, which is J1, is B goes to A at a rate that is, as James was explaining, mass action is proportional to B. For the parameters, now that I have defined these parameters here, K1 and K2, I have to specify what their values are. And I'm gonna set them to be 0.01 and K2 0.025. It has to be colon. Then my initial conditions, I'm gonna set them to 10 for A and no B. And I wanna make sure that everyone is with me here. Okay. So some people do need more time, so I'm just gonna wait a couple of minutes. But just as a recap, we had created a basic simulation where we've added two types of cells, cell proliferating and quiescent. We assigned them the contact and the volume constraint. We've defined a basic antimony model that describes a reaction where A goes to B at rate K1 times A, and B goes to 8 at rate K2 times B. Okay. So that describes the model for us, but this is just a string at the moment. It's not interacting with CompuCell at all. It's just a string that we have specified in the step hour. Now we need to be able to load it as a free-floating antimony model. And again, when we talked about a free-floating antimony model, it's a model that is not attached to any object of our simulation. It's free float, and that's why it's free floating. We can still, CompuCell is still gonna step the model, and we can still interact with the model, but the model is not attached to any of the objects of our simulation. And if we want to load that model, we always do them in the start function. And to have access to the code that would allow us to load the model, we always come to your friend CC3D Python, and we go to SVML solvers. It's a little bit of a long list here, but the ones we are interested in is this add free floating antimony model. There's many options depending on the type of markup language or model specification that you're using to specify your subcellular model. But in this part, as I mentioned, in this particular exercise, we're gonna focus on antimony models. So we add this free floating antimony model, and then it has a, let me decrease it a little bit. So it has a little bit of information for us. It says that if we have a file, we should name that file model file and have the path to that file. So if we have the antimony string in we haven't specified it up here in our same step hour, we could have it as a independent file. save it in an independent file, and we could load it that well, reference it that file, or we can reference directly the model string. And it tells you what the structure of that basic structure of that model string should be. So if we look at the options that add-free floating antimony model has, as I mentioned, there is a model file. If we have a path to a file that contains our antimony model, but we do not because we wrote our own model, our model string. We already called the model string, so this serves as the input to as this input to the function. Then we have to come for a model name, and this part is important because the model name is what we're gonna use to reference the model later when we want to retrieve, write and read the data or values of our or any parameter values or variable value, state variable from our model. So for this particular example, I called it my model. Step size is another important concept, and it refers to how many the basic time step in CompuCell, as it was discussed some time ago. It's called the Monte Carlo step, or what we call the MCS. Now we have to specify, and this part is a little bit too complicated, but for now we're just gonna set it. We're just gonna describe it shallowly by saying that the time step specifies how many of the internal units of the model corresponds to one Monte Carlo step. For now, we're just gonna set that to 1. But this becomes very important. So 1 unit in Monte Carlo step corresponds to 1 unit in the intracellular model. So let's say my Monte Carlo, I said I arbitrarily decided that a Monte Carlo step is equivalent to 5 seconds. Then I'm saying that each unit, each single unit in the subcellular model is also corresponds to 6 seconds. But this becomes very important when you want to not just integrate, but also synchronize the units of your tissue level model with the units of your subcellular process. For initial conditions, we're not going to specify the initial conditions because we have already specified them in the antimony string. And then that way, we have already loaded our antimony model. So if we run this, should be able to run, although I'm not into CompuCell is solving the model, but I'm not doing anything with it. So I'm not gonna be able to see anything really, just my cells. So I can see my cells. Right, but there's really nothing interacting with them. And I haven't, although CompuCell is actually solving the SBML model, I have no way to see how that is sort of like hidden at the moment. So what we will do is to be able to see how CompuCell is solving the SBML model is that we're gonna plot the values of the state variables of my model. So that's the next step. Oh, I forgot. Actually, I made a mistake. A mistake that I did. I mentioned that it was very important, and I absolutely forgot. So, any ideas of if I ask the question now, is CompuCell3D step in the model or not? What is the answer? No, it's not, because I forgot to actually call that important function that it's supposed that I am always supposed to call when I'm using SBML models to step the model. And TJ, I'm sorry, Tarim has an important question. I will let TJ answer that question, but I'll just continue with this while TJ can explain that in the chat. So I forgot to time step my model. So if I want to get the code, I can type it because I know it already. It's not that complicated. But if I forget it, I can always come to CC3D Python, SBML Solver, and then there is this time step SBML models that allows me to time step the model. And now CompuCell is time stepping the model. And if I rerun CompuCell, it's just gonna be the same because I'm not interacting the model. But in this case, now CompuCell is actually time stepping the model. And I'm gonna relaunch the poll just to make sure that everyone is with me up to here. Okay. So yeah, most people seem to be with me here. I'm just gonna continue on. The next step is that we actually want to be able to see how CompuCell is stepping the model. And to do that, what I will do is we will create plots and we will retrieve data from this free-floating model, and then we will add it to our plots. And this should be, by now we have done this a couple of times, so I'll go fast over it. For scientific plots, we always come to CC3D Python, scientific plots, setup. I called it free floating model variables. I turn on the grid just as an exercise to show that you have some ability to modify the options here. And then I created two data series because I have two variables. One is A, the other one is B. And then I want them both to be dots. And then red and green is fine, or red and blue. I call them different. I had a different setting. So that it's adding the plot window. So remember CC3D Python, scientific plots, and then I just rename the title of my plot, and then I created two data series, A and B, such that I can add the data from the variables A and B. I run the model now. I should get a window, an additional window that it's my plotting window. But if I run the model, I shouldn't see anything, right? Because even though I created the window, I haven't passed on any data. To pass on any data, I have to do that in the step function. So now, and this part is important, I'm gonna add the data points to the plots. And to be able to do that, I need to be able to retrieve or reference the model that I've just loaded. So I'm gonna create two variables, A and B. And on A, I'm gonna read the value of the variable A in my model. So the way I do that, I reference that is by this self SBML my model, and then the name of the variable. That I type directly because it's very easy. But again, if you do not know what the reference is, again, you can always come to SBML model, and then you get the SBML value for a free floating model. And that is supposed to give you the same syntax. When I use it here, and see that now where it says model name, I have to change it to the name of the model that I specified when I load it, and then the variable that I am interested in reading. And now that I have, now every time step, I'll read the value of the variable A in this free floating model. The next step is adding this variables to my plots. And I always come to scientific plots, add data points. These ones, since are a little bit long, I do not type themselves manually all the time. But I know that now the data, I have two data series, A and B. And then I store the values of the variables in the free floating model in these variables A and B. And now if I save and relaunch my simulation, I should be able to see the so Farnoosh, I did it really quick, but when you loaded the model, there is a variable called initial conditions that I deleted because I have already defined the initial conditions in the Antimony model. So you can delete that part of the function or the class instantiation. So yeah, so now I'm able to plot the model. And the reason I included the cells here is because I wanted to show that the cells are in this free floating model. I created the free floating model, but I really haven't done anything with my cells. So the model is basically being stepped and resolved by CompuCell, but it's not interacting with myself in any way. So the simulation shouldn't be affected by what is happening in this free floating model or my CompuCell simulation. Okay, I'm gonna relaunch the poll just to make sure that everyone is with me. So so is there any, Polly, there seems to be, maybe there is a problem with your model specification in XML. So I'm gonna, if you need, is there anyone available to help in a breakout room? In the meantime, I'm waiting on other people to catch up. Okay, so So this is basically the same code you had in your previous it is not in the medium, right? It is global in the sense that it's not in all cells. It's just independent of every object in our simulation. So basically, it's running parallel to my simulation, but it has no interaction whatsoever with my simulation unless I specify it. But it's not in the medium. It's not in any of my cells. It's not on all of my cells. It's basically nowhere. And that's why it's useful to model processes at larger than the tissue scale, because this will happen basically, let's say above my or in the scales up or above my simulation. So that's why that free floating is a good term to think about it. So I'm sorry because I'm also assigning to breakout rooms. So okay. Okay. Well, so as I was mentioning, this is basically the same simulation we were playing with in the previous module. It just had the option for you to specify your antimony model, and then it was plotting the state variables of your of the model specifying the as a free floating model. Let's do something a little bit more fun. I'm sorry, I do not see. Right. So now we can show, now that I've read from my free antimony model, I can actually also write to my free floating antimony model. And the syntax will be basically the same. So let's say that at time MCS 400, I want, for whatever reason, my variable A or the value of, I'm sorry. I'm using a Mac on Nano Hub, and then I forget that they're not the same keys. So let's say that at time 400, we want our model, we want the value of our free float, the value of A in our free floating model to change. So what we do is that we use the same reference, but now we set the value instead of reading it. Okay. And then now I can save it's as easy as that. And then if I rerun my simulation, I can see that it's CompuCell is stepping the model, but when it reaches 400, then the value of A changes, and consequently the value of B will change as well, of course, because there is an interaction between A and B. So the equilibrium concentration of the two variables changed. So For those of you who need this as a reference, this exercise have been uploaded as exercise 411 in the Google Drive. Okay, I'm gonna give a couple of minutes for the people who went to the breakout room to come back, and for people who are still working on the exercise to catch up before we move on to the next exercise. In the meantime, if anyone has any questions, please feel free to ask.
Can you show your code again?
Yes, 1 second. So Ben, I'm actually not exactly sure. I think that's a little bit of leftover from previous version, but I think the state, you can reference the state of the model.
So I think state is just referencing the whole model.
Oh yeah, yeah. So it's how to reference the model, right?
Yeah.
Because if you look at the syntax that it provides, it's exactly the same syntax that we use to reference the model plus the variable of interest. So it's basically returning the model or the state of the model as an object. Technical terms. Okay, I'm gonna continue with the next couple of exercises. In the next, managing the Zoom meeting while presenting, it's actually more complicated than it seems. I have like 5 different windows all on top of each other. So in the next exercise, in the previous exercise, we loaded the Antimony model as a free-floating model, right? And I explained a couple of times that there is particular uses for that type of model integration. But if we want to model subcellular processes, the more natural way of coupling our CompuCell simulation with our models is to load this subcellular models into each of our cells. So this is what we will do in our next exercise. We will just continue building up on this same code. Yeah, sure, let's do it. Let's continue building upon this code. We're just gonna have windows that we're not gonna be using. So the syntax to let's stick with the same antimony model, but the antimony model now we're gonna interpret as something that is happening in a reaction that is happening inside cells. So actually, please follow me what I'm doing next, because we're not gonna be able to use the plotting. Please delete the syntax that we use to create the plotting windows, and the syntax that we use to add data points and to reference the values of the SBMM model, of the free floating model. So basically, we're just gonna leave the time step function inside the step function. And then let's go ahead and delete everything from the start function as well. So basically, to start this example, let's just start with a blanket start function and then a step function that just has this time step, the time stepping of the SBML models. So let me just make sure that everyone's with me there. So I didn't do anything fancy. I just deleted everything from my start function and basically everything from my step function. So I can use this as a blanket simulation instead of having to create a new one. As I was mentioning, we have the option of loading this antimony model that we have specified inside each cell or inside a cell, depending on if we interpret the model as a reaction happening inside a cell, whether it be a signaling process or a gene regulatory process, or even a metabolic process. So when we interpret them that way, then we can the most natural way of integrating CompuCell with those models is to load them into the cells. And to load the model that we have already specified into the cell, we just come back to CC3D Python again, SBML solvers, and then we have three different options here. We can add the antimony model by cell ID. We can add it to by cell type, or we can add it to individual cells. And we will start with adding it to a cell ID, to a model. We will start with adding the model by cell ID. Again, I get the code snippet and it looks very similar. It's just changed. Instead of add free floating antimony model, it now has add antimony models to cell ID. The model file, like explained already, we wrote it, or we wrote the model string, so we don't have to use that one. The name of the model, let's again call it my model. And let's just model to cells that have ID 1, 3, and 7. We have more than 7 cells, so we won't have a problem. But this is basically, the cell ID is the identifier of each of the cells in our simulation. It's a number that identifies each of the cells. It's basically the identity number. So in this particular case, I'm gonna upload this model into cells 1, 3, and 7. Step size, let's again make our step size being 1. And again, initial conditions I specified in my antimony model, so we can get rid of that part. So all I need is the model string, the model name, and the cell IDs and the step size. So good question. You see that this is a this is a list, right? The data structure is a list. So cell IDs is expecting a list as an input. I could pass a list in that particular case that goes from 0 to, let's say, 100 or 1 to 100. And that would generalize it to all of the cells with ID from 1 to 100. But we're just gonna stick to 1, 3, and 7. Okay. So what we will do next, I did all of the options at the same time, but we're not gonna do that now. What we'll do is that we are gonna, we want to be able to visualize the state of the subcellular model in all of our cells. There's multiple ways we can do that. We could potentially plot the value for each of our cells, but then if we have many cells, that would be a lot of plots on top of each other. That might not be meaningful. We could plot the average of the values. That's fine as well. But in this particular case, because this stage is pretty exploratory, what I'll do is I'll just plot them visually, so I can see, I can color my cell depending on the value of the particular variable of interest. So to do that, I have to create a auxiliary field. We did this already yesterday, but we're gonna review it. To create, and remember that again, the auxiliary field, what it does is basically creates a window in our simulation in player where I can display the value of a variable inside a cell depending on the value specified in the dictionary of that cell. So to create this auxiliary field, the first I always do it in the init function. I come to CC3D Python, extra fields, automatic tracking, track cell attribute. And we're gonna track two. We're gonna track the value of A in our cells and the value of B in our cells. So we need two of these fields. And let's call it the field name. Let's call it A and B. And the attribute name is a reference to, yes, it's CC3D Python extra field automatic tracking track scalar cell attribute. And this has to be done in the init function of the step hour. And then the attribute name has to reference the attribute in the cell dictionary that I'm interested in displaying. We haven't created them yet, but we will call them A and B. So let's just call the attribute name A and B. So the field name is A and B. The attribute names is also A and B. But now I have to create and update this attribute in the cell dictionary. And that is just as simple as in the step function, adding the attributes, I'm sorry. A and B to my cell dictionary. And then question for you, how do I reference the values of the subcellular model yes, I'm sorry. Yeah, I have, right, cell is a reference, so it needs to be let me see. Right. I'm sorry, I created, okay, yeah, this is just a little bit more complicated that I wanted to do it, but I'll just do it in the step function. And I'll set them to 0 for now. And then I'll explain how do I reference them later. So let's, and I'm sorry for the confusion here, but what I'm basically doing is each step I am iterating over my cell list, and I am updating the value of this cell dictionary attribute called A and B, and I'm setting it to 0. If I come back and I run the simulation now, now I can see that I have the fields A and B. If I run the simulation, there's nothing is going to happen because I haven't tied up the intracellular models to the values of these cell dictionary attributes. But I've only created the cell attributes. I'm gonna make sure that everyone's with me. I'm gonna put the code back. So the people who need help, feel free to ask questions here, or I can assign you to a breakout room if preferred. Okay. Is anyone available to help Tarim? Okay, I'm gonna assign to your breakout room. Okay, good. So, and this is why I wanted to give people time to catch up, because this part is a little bit complicated and I do not want to get into the details here, but the basic idea is that we have just loaded the antimony model into some of our cells, not all of our cells. We have loaded into cells number 1, 3, and 7. If we try to reference the antimony model on a cell that does not have one, we're just gonna get an error. So the way my way around it was to use this exception handling. And I'm not gonna go into the details. So we actually good question, Polly. Good practice is always to initialize the variables that you're gonna be using in your simulation. Excellent point. So, right. But really, if you think about what the flow of the flow of the code is, this step function is called when Monte Carlo goes from 0 to 1, and if the dictionary did not have a attribute called A, it will create it at that time, and that's why I didn't create it in the start function. But yes, it's always good practice to create the variable, initialize it in your start function. Okay, so back to this. So again, what I was trying to explain is that we haven't loaded the antimony model into all of our cells, and if we were to try to if we were going to try to reference the antimony model in a cell that does not have it, we will our CompuCell will issue an error. So I did some exception handling here. I'm not gonna go into the details. This is a little bit more Python-y than CompuCell. But what I'm basically saying is here, if the cell does not has a antimony model, just keep going, do nothing. But if the cell has an antimony model, then I want to update the value of the dictionary with, right, we could also do that, but I generalized it because of the other exercises that we're gonna have later. But yes, you could actually iterate through cells with ID 3, 1, 3, and 5. There's many ways you could handle this this complication. But now the question I wanted to ask you is, now that I have handled that problem, if I want to update the value of the dictionary attribute A, how do I reference the model inside that cell? I mean, the answer is there, but I also wanted you to give it a try by looking at the CC3D Python. So we're gonna reference the model inside each cell by typing cell.sbml, the number of the model. That's not the format. Note, in the previous example, when we loaded the model as a free antimony string, do you guys remember what the reference was? If not, can someone pull it from their CC3D Python pull down menu and type it in the chat? What was the reference for the free floating antimony model? Right, it was self.sbml model. That references any free floating antimony model. If we want to reference the model inside each cell, then we had to get the reference to the cell and then reference the model inside that cell. So now we can reference the values of the variables inside the cell. And then when we come to CompuCell and execute the simulation now, we can actually see what the value of the variables A and B are inside each of the cells that I initialized the SBML model. The reason they look both red, again, it's because my visualization options. So I can come back to my visualization properties, and I know that A and B can only go to 10. 10 is the maximum value they can go to. So I can readjust the color bars. So I can see the relative values of each variable in the cells that are initialized the SBML model. And I'm gonna relaunch a poll just to make sure that everyone is with me. Good. So this exercise is available to you as exercise 412 in the demos. The same way we did with the, so we loaded the, model. So we've loaded the SBML model as by cell ID. In the example that and then I have all of the examples of what it looks like when you load them by different options. We'll try some of them to see how the syntax differs from the one that we've used so far. So another option that we have is to load the Antimony model by cell type instead of cell ID. And to do so, we come to CC3D Python SBML solvers, add Antimony model to cell types. And again, I have to fill up what the function, I have to, the function inputs, I need to fit them correctly. Our model string, our model name, we're gonna rename that the same way so we can reuse the code. And then now I have to specify instead what the cell types that I wanna load this model into are. And you might recall that we created two different cell types. We call them proliferating and quiescent. Let's just load our model in our proliferating cells. And we have to type the cell type all in caps. And again, we have to specify what the step size is, and let's call it 1, and let's make it 1, and initial conditions we can get rid of because we are specifying them in the Antimony string. Now, if I come back to the simulation and I reload the simulation, so I can see that blue are proliferating, green are quiescent. And if I look at the value of A, it's only loaded in the proliferating cell type and not in the quiescent cell type. Relaunch the model and show the code. some people, there seems to be a decent amount of people that need time to work on this. So I'm gonna just give a couple of minutes to people to catch up. Again, I want to remember everyone that this exercise are available for you in the Google Drive, and it's called exercise 4112. And while we wait for everyone to catch up, I will be happy to answer any questions. I'm not sure, Ben, I'm not sure that it's a satisfactory answer to that question, except that maybe there is a particular reason why Juliano might know, but I actually might think that it's the way it was done at the beginning and it's the way it's been done since. Right. The end of line 33, 36, model string, the model name, mile name, and then the cell types that I want to attach those SBML models to. Right, Joseph, if you, right, because I fixed the values of my color scheme, they look teal and cyan because I fixed the values to be 10 and the actual numbers are lower, which is like equilibrium numbers that we saw earlier. If it's automatic scaling, always the highest value is going to be red and then the smallest value is going to be blue. So they will both look the same. The reason I fixed the scales is because I wanted you to be able to relate the previous values in the equilibrium concentration where A was lower than B to the colors that were shown in the visualization windows. Okay, so so I do want everyone to catch up with me because it gets more complicated from here on. So I'm just gonna relaunch the poll to make sure that at least most people are with me, or at least are just watching the presentation before moving on. Or so you can click yes if you think it's time for me to move on to the next exercise. Okay, so there is, as I mentioned earlier, there is many other options that we can use to load the antimony models. I'm not gonna do them all now because it's a little bit repetitive. There's more interesting examples to work with, but I'm just gonna let you play with it as a homework. You can try loading the antimony model to all of my cells. And for that, what I do is that I loop over my cell list, and then I use this syntax called add antimony model to cell. And then when it requires me to load the cell, I load it to the cell that I'm referencing. There is a typo here, actually. It shouldn't be cell 20 that's just because I copied from the previous line. It should be cell, which is the reference that I'm using to the name I'm using to reference each of the cells in the cell list. And that will do is that will load the antimony model into all of my cells. So there's a little bit of a subtlety there, but the idea is that if I want to load it into all of my cells, because it's a process that I think happens in all of my cells, all I need to do is to loop over my cells and then load the model in each of the cells. Okay, so now let's move on to a little bit more biologically relevant exercises or examples. In the next session, in the next part of this module, we're gonna see how we can use the principles discussed up to now to model delta notch, and whether we can reproduce the patterning characteristic of the delta notch signaling. And we will show how we can actually add steering panels that allow us to change the parameters of our SBML models on the fly. And again, James was talking earlier about how CompuCell can be used in two rather distinct ways. One way of using CompuCell is, which we tend to use at the very beginning of when we're building models, is exploratory. And in that, when we're using CompuCell in an exploratory mode, we tend to rely a lot on the visualization, on how the simulation looks like. And then steering the parameters on the fly allow us to identify what parameter values better match the expectations that we have in terms of what the simulation should look like. Once we've settled with the parameters and the mechanisms that we've incorporated, we move on from there and we rely much less on the actual visualization. But for the exploratory model, we do rely a lot on this point, on the visualization and the steering. I'm gonna go very quick over delta notch. I'm not an expert on delta notch, but just as a quick introduction, delta notch is one of the most important mechanisms for cell signaling. And notch specifically is a transmembrane receptor that coordinates and activates the signaling through the notch pathway. And the notch signaling pathway is associated with different downstream effects in cells, including the regulation of sulfate and formation at a tissue level of pattern, If you want to have a better idea or more get a better review of the delta notch signaling pathway, there is a nice reference here. In terms of molecularly, what's happening in the delta notch, in the delta notch is that it's acting as a molecular switch. Both delta and notch are transmembrane proteins. Delta is the ligand of notch. And then delta works as a signal from the signaling molecule onto the signal receiving cell that has a notch, or where notch is activated. Sort of trying to keep track with the chat. So, and then once notch is activated in the receiving cell, then this activation leads downstream to the inhibition of delta in that same signal receiving cell. So basically, delta from a neighboring cells activate notch in the delta in the receiving cell, and then the downstream effect of the activation of notch is it inhibition or downregulation of delta on that same cell receiving cell, signal receiving cell. And that leads to this usual pattern formation where we see high notch, high delta, high notch, high delta, high notch, high delta, or high notch, low notch, high notch, notch, low notch. There's been a couple of mathematical models explained, proposed to model this delta notch activation. This is we're gonna for this example we're gonna use this model published by Collier, and it's a very simple model. It just have two differential equations, one describing the activity of delta and one describing the activity on notch in a single cell. I wanted to quickly explain how to interpret these equations. It's a little bit beyond the scope of the class, but just as a quick readout, once I see these equations here, the functional form of these equations, I immediately interpret them as activation or inhibition. And that is what this is doing. So we can read this as delta activating notch, or this delta bar activating notch, and then notch inhibiting delta. And delta bar is a parameter, it's not a variable, it's a parameter of the model, and represents the average delta signal that a cell receives from its neighbor. So remember that this two equations, sorry about the exterior noise. These two equations are describing, and this is important to remember, the state of the delta notch signaling inside a particular cell. In a particular cell, how much notch activated there is depends on how much delta is in their neighbors, which works basically as an input parameter in my in the delta notch model, and then the state of the notch variable determines the amount of inhibition on the delta variable. Okay, and that's all, and with that model we can actually, simple model, we can actually reproduce the special patterning of activation on activation inhibition that we see in the delta notch signaling. So what we'll do for this example is we will create a very small simulation domain, just one single type of cell. And we're gonna assign them both volume and contact constraint. And we're gonna fill the whole simulation domain with our cells, so there would be no open space. And then we're gonna write an antimony model that corresponds to the ODE equations that we just saw. James discussed a little bit of that earlier. We're gonna put that in practice now. How do you go from ODE equations to the antimony string? And then we're gonna load the antimony string into all the cells and initialize the value of delta and notch. And then we're gonna calculate what this delta bar or D bar represents for each individual cell. And then we will again create auxiliary fields that are gonna help us see the level of delta and notch in all of the cells that we created, okay? So we're gonna, okay. So we're gonna build the simulation together from scratch. So CC3D project, new project, let's call it delta notch. And I said it's gonna have 42 dimensions. And actually we want our boundary conditions to be periodic on X and Y. Our membrane fluctuations of our pixel copy range, not our membrane fluctuations, to be range 3. And we want the uniform initializer, which is a rectangular slab. We're just gonna create one type of cell. Let's call it my cell. No chemical fields. And I said that we will have contact and we will have volume local flex, so we can specify the volume constraint parameters in Python. And we're gonna finish the simulation. So let's make sure that everyone has a blanket simulation to work from. The plugins are volume, local flex, and contact. Okay, I think everyone is with me now. So let's close this exercise 1. Let's open delta notch. And so in the XML, we should have the dimensions of our simulation, the boundary conditions on X and Y, our 2 cell types, our cell type. And then we want to create, we want to fill the whole simulation domain. So we're gonna go from 0 to 42 and 0, I'm sorry, I always type them incorrectly. 0 from 42, 0 to 42 on X and Y. So we can fill the whole simulation domain. I didn't do anything here. Let's just launch it to make sure that that we have a working simulation, and that it's filling up the whole simulation domain. Yep, it is. Okay. So let's make sure that everyone's with me here. Yeah. Okay. So yeah, pretty much everyone is with me now. So in the next step, we're gonna have to turn our antimony model with our antimony model, our ODE into an antimony model. And I'm gonna ask this question so so you can answer in the chat. How many of you want to have time to work that out yourself, as opposed to me doing it for you? So I was thinking on giving you time to. It's not complicated, but I wasn't sure if you prefer me to do it right away or to give you time to try to figure it out yourselves. Okay. So let's do it together then. So, and this builds up on what James was explaining earlier. So there is a way we can, there is a simple way to return, well, I mean, knowing the syntax of the antimony string would allow you to turn this ODE equations into an antimony model. The first thing is we want to define the model string. We haven't defined them yet, I'm sorry. And that is a good point because we were supposed to define them in the start function. So in the start function, yeah, we said that we will, as for the target volume, we'll set it to 50. And then the lambda volume, we'll set to thank you, Polly, for reminding me. So now we can come back to writing the model. And I'm not gonna spend a lot of time into doing it, but again, I always have the, it's always good to comment on your string so you know which part corresponds to what. Divide it into your equations, your parameters, and your initial conditions. Now, in these equations, notch is not becoming delta, notch is regulating delta. And the way we end, same with D bar is not becoming notch, D bar is up-regulating notch. So the way we write those types of regulatory interactions is by, we call it nothing going to the variable. So it's gonna be n and going to be d. So this is basically the up-regulation or inhibition of notch and delta. We call it also nothing goes to n and nothing goes to d. And the regulatory parameter for notch is gonna be d bar over d bar plus a, and the inhibitory interaction in delta is V times one over one plus beta times n to the h, right? So the regulatory interaction on notch is d bar over d bar plus a, and then the inhibitory interaction of notch on delta is b times 1 over 1 plus b plus n to the h. Let's make sure that everyone is with me here.
There is minus d and minus n there in the equations, but
Yes, we will type them now because we were first typing the first terms, and those we could directly type them as minus d and minus n, but I also wanted to show that since they represent decay, we could also type them differently. It's equivalent. So let's go ahead and do it now, and then I'll just explain what the equivalence is. So in n, we have a decay term that is called n, and I can just type minus n. And then in d, there is a decay term that it's d times v, right? So it's inside the parentheses. But yeah, as I could have typed them as well this way. And those two, this and this, are equivalent to this part. So they are doing exactly the same function. Just gonna delete them from there though. Or I can leave them and comment them out, but I'm just gonna delete it to avoid confusions. Okay, now I have to type the parameters. A. It's gonna be
if the equation
0.
is kind of second order, can we write in this antimony in this part? If it is the second order, the equation, is it possible to write?
I mean, if it's a second order, like in n, like
D,
like in
D2D over
right.
D2.
Right. Yes. Yeah. The specific, like if this is, for example, the easiest way of just taking the ODEs and writing them up here. So you could potentially do it as well. Like let's say it could be d square here, or n square here.
I mean, in the derivative term in the first part.
Here.
In the equation in the left part. Like the second order derivative, I mean.
I'm sorry, I did forget this part. Is this what you're talking about? Because I did forget to write this. So it's d bar to the k, and then n to the h. So, but yeah, I'm sorry, go ahead and ask your question again.
I'm just talking about the second order ODs to model it like that. Is it possible?
So so can you type an example in the chat of what the
Okay,
equation
okay.
will look like? What type of equation do you have in mind? And yes, for people who are asking, I did forget to type the d bar to the k. So yeah, I do want to answer your question. I just didn't understand what the, yeah. Oh, if it was, okay. Right, I'm not exactly sure. That's an excellent question. I think there's people in the group in the seminar that are more right. Now I do understand. My guess is gonna be yes, but I'm not exactly sure because I already have a reference to the rate, right? So I could potentially reference the rate in my equations, but I'm actually not sure. That's an excellent question. So if I make it as a separate reaction. But again, yes, I mean, I would have to, I could. If I type it this way, right? The rate here, I can type whatever I want. I can make it d squared, or I can reference, let's say, I'm guessing, but I've never tried it, that I can potentially reference the rate previously defined or any other functional form that I want. I can define a function and then use it here. So I'm free to specify the rate at whatever rate I want. But I'm not necessarily sure if I can actually reference the rates themselves for, let's say, second derivatives or second order derivatives. But there's people who are more of experts in Tellurium in the chat that might actually be able to help with that part. I'm actually not sure if I can make my functions, my rates depending on other rates, for example. Yes, there is a way of changing the values of the model parameters, just the same way we have referenced the values of the variables and we have set them. You use exactly the same syntax, but instead of referencing the values themselves, you then reference the parameters. So let's just continue here. Again, questions that I might not be able to answer directly. There's James is coming. He'll probably be able to answer, and there's also other people in the group that knows in the chat that know better. So let's continue specifying the parameters of the delta notch model. And then for the initial conditions, I'm just gonna set them to 0 here because I'm gonna initialize them elsewhere. So this would be the antimony equivalent in, I forgot K. K equals H, right? K equals 2. There you go. So this would be the equivalent antimony string of the ODE equations displayed here. I call the average in the example, but it's the same as D bar. Yeah. So let's call D bar. That's another good question. D bar, let's set it to 0.4 into a 0.1. Right. Thank you everyone for reminders. I think I'm just gonna need a break here soon. So making a lot of mistakes. So but let's just try to get as far as we can in implementing the delta notch. So yeah, this is the equivalent. D bar is a parameter in this model, right? Because how do we know that D bar is a parameter? Because there is no rate equation that describes the change of D bar. So that it's interpreted by the antimony parser as D bar being a parameter. Okay, and then in the next part, we're just gonna load our antimony model into all of our cells. And we do that in our start function. Again, CC3D Python, SBML solver, add antimony cell, let's say to cell types. Let's use the cell type syntax. And we're gonna load it into we're gonna again fill up the inputs on that function. I actually do not remember how I called the model string. I called it model. Let's call it model string. And let's call the model name is gonna be DN for delta notch. And then the cell types that I wanna load it into, if we remember, the cell type we created is called my cell. And then for the step size, we will use here 0.2. And why 0.2? It's because I wanted to slow down the frequency at which the signals was being updated in the cells. But that's just, I mean, that's a little bit more advanced. It doesn't matter for the purpose of the module what the step size is. So it's a matter of how much resolution do you want. And again, we always want the integrate function or the step function inside the step in our step our CC3D Python SBML solver time step SBML model. So that will load the SBML model into our cells, and then yes, Polly, we will later update dbar reading information from the simulation. But for now, we just have to initialize it with some value. So we just put a value there, but we will update that value by reading the delta in our cells later on. And that's how we're gonna turn the delta notch model into a multi-scale model. But let's just, so this part finishes the loading Yes, I did. No, I'm sorry. No, I did not. It's outside because I'm loading it by cell type. So I don't want to load it. I just need to load it one time. I don't need to load it every time I iterate over my cells. To call time step SBML, CC3D Python, SBML solvers, time step SBML model. Well, welcome back, everyone. Let's continue with our previous exercise where we were building a multi-scale model of delta notch signaling. So far, we've defined our delta notch model from the ODE equations. We have specified what the parameters of that model are and what the initial conditions. And we've loaded our antimony model in all of our cells. In the next exercise, as some people pointed out already, now we have to tie one of the parameters of our model, the delta bar, the D bar parameter, to some of the properties of our CompuCell simulation. And in particular, the way we're gonna tie this D bar to the CompuCell simulation is that we will, if we have a single cell, we will look at all of the neighbors of that cell. We're gonna see what the delta value is in those neighbors. And then we're gonna average over the neighbors of the cell. And that's gonna be the value that we pass to D bar. Again, just to make sure, because this part is really important. The way we're gonna calculate D bar inside each cell is that we're gonna look at each of the neighbors of the cell, see what the delta value is in each of those neighbors, and then we're gonna average that delta value over the neighbors. So we're gonna have an average delta from their neighbors and pass it on as a D bar to that cell's delta notch model. Does that make sense? Want to make sure that makes sense because it's really important. It's subtle, but it's actually the way in which we are building and the multi-scale aspect of the model or of this simulation. Anyone need me to explain it again?
I would appreciate you explain again. Thank you.
Okay, let me see if I can draw because that's probably better. Yeah, I do. So we have a cell, right? And then inside that cell, we have a delta notch model, right? So we have a value for D and we have a value for N in that cell, inside that cell. And that cell has multiple neighbors that also have a DN model inside. And all of the model have the same structure in the sense that they all have the same equations and the same parameters. One of the parameters in this DN model is the delta bar or D bar, the D bar parameter. And the way I'm gonna connect or make this cell multi-scale or this simulation multi-scale is that the delta bar in this parameter, in this cell, I'm gonna calculate it by looking what the delta value is in each of their its neighbors. And then the delta bar is gonna be the average delta of the neighbors, average delta. That's what the D bar is, the average in the neighbors. Does that make sense? So each of my cells has a single instance of the delta notch model, which means that each of them has a D and an N, a delta and a notch. And then the way I calculate the parameter D bar for each of those cells is that I look at their neighbors and average the delta of their neighbors. Okay. So the way I do that is, as is explained in this code, is that I'm gonna, in the step function, I'm gonna iterate over every single one of my cells. Then I'm gonna create a variable that I'm gonna call the average, set it to 0, and a counter, and set it to 0. And then now that I'm at a particular cell, I have to visit or get the reference of each of the neighbors of my cell. And if I want to do that, there is a convenient Python script that allows me to do it. I go to CC3D Python, visit cell neighbors. Okay. And then this for loops is looping over this get cell neighbor data list, which is basically the list of the neighbors of a cell. And if the cell has a neighbor, right, then I will do something. And what I'll do is that I will add the delta value in that neighbor to my the average value variable. And the way I reference, right, I define a D bar inside the SVML model, but that's independent. I will have to, and later on, I will make the equivalence between D bar in the SVML model and the average here in the Python code. But I named it differently at this example just to make it clear that they are different. They have different functions. So the average what I need to do now is I need to get the value of the delta in the neighbors. And neighbor here is basically has the same function as cell in this loop, except that it references, instead of referencing the cell, it references the neighbors of a cell. So I can use this neighbor reference the same way I use my cell reference in the loop to read the value of the delta in the cell in the neighbor. And then after I added the value, then I increase my count on my counter by 1. And then at the end, after I iterated over all of my neighbors, I can divide the average, I'm sorry, by the number of neighbors. SBMLDN is the reference to the SBML model that we called DN and that we loaded into each of our cells. Okay, so in the previous part, we defined the antimony string and we loaded into all of our cells. And when we loaded it into all of our cells, we call it DN. So each of our cells has a DN model. And then what we're doing here is a little bit complicated, but the idea again is that we are the idea here is that we are basically averaging the delta value of the cell neighbors. And counter is just I'm doing counter to know how many neighbors that cell has. So every time I visit a neighbor, I store the value of that neighbor's delta, and then I increase the counter such that I can then average the delta over the number of neighbors that I had or that I visited. Okay. Right. So I added if counter because let's say the cell has no neighbors. I want to avoid a division by 0. So I added the if counter just in case my cell has no neighbors, my counter is 0. So I do not, the average remains, that stays 0 and I do not divide by 0. Ying, that's an excellent question. I always have, TJ or Julian are probably better to answer that question. What range does the neighbor function iterates over? And I'm pretty sure we've tried to figure this out by.
What
Yeah,
do you
go
mean
ahead.
by the neighbor function? There's a setting for that.
The for neighbor loop is what we mean. When we get the self get neighbor data list, whether that includes the corner neighbor. It's basically asking what order the neighbor visits over.
Right. It should be set by the lattice definition.
Right. But I am actually not 100% sure about that. I think it should be, but I'm not 100%. That's why I'm passing on the question. Let me launch the poll just to make sure that everyone is with me here. It's also important because this is actually the key part of the model. We haven't done anything different from what we've done in the previous exercise until this part, where we are trying to connect the individual delta notch model in a single cell with some properties of their neighbors in the CompuCell simulation.
Could you explain a little bit about the for loop that you write here? And you said neighbor, comma, common surface area. These two, there are two variables there in
Right.
the for loop, right?
Right. So when Python allows you to iterate over 2 different variables at the same time, as long as you're iterating, let's say, over something that can assign values to both of them. And then this cell neighbor data list gives you the reference to the neighbor. That's what we're calling neighbor here. It's the reference to the neighbor. And common surface area gives you how many pixels they have in common. How many pixels of your cell are in contact with the neighbor.
Okay, so
Does that answer your question?
Yeah. So, but you didn't use common surface area in the loop, right?
Right, I did not. I didn't. I mean, that's also something we could potentially do. We could scale the delta value by the common surface area. That is a little bit of a different way of connecting it, a different way of thinking about how to connect the models, right? In this case, I just took the raw number of delta, regardless of how much common surface area they have. But in principle, right? Since delta is a soul surface receptor, it would make sense to scale my delta, how much delta I read off from my neighbor, by how much common surface area I have with that neighbor. So that's an excellent question.
So if you delete the common surface area from there, does it going to have an issue?
I don't think it works because the get cell neighbor data list is
Two outputs, right?
right. It gives you two objects.
Okay, got it.
Right. So that's why Paulo is explaining. The function returns a tuple, so you need to iterate through it with a tuple as well, with a tuple as well. Cool. So again, the idea here is this is an important part of this code, because it's a part that is allowing me to connect the delta notch in one cell with the delta notch in a different cell. So now that we have calculated this D average, all we need to do is to update in the cell the value of what we call D bar based on the average delta over the neighbors, right? So I am already iterating over my cell. So all I need to do is to now reference the delta notch model inside my cell and override the value of what I call D bar to be the average. Okay. That part is important as well, because in the first part, I just calculated the average delta of the neighbors. And in this part now, I'm overriding the D bar parameter inside the delta notch model with that value that I calculated in the upper part. Okay. So now, so Giuliano is pointing out that even with neighbor order or the pixel range 2, this function is always returning first order neighbors, which means that the ones that are in contact with the edges of the cell, not with the corners. Yeah, I wrote it a little bit different in this code than in the code that I'm showing in the as an example. Just a matter of writing it 2 times, but yeah, that is equivalent. That code that I had was just equivalent to doing this. It's just it is equivalent. Okay, so now now we have actually finished building our delta notch simulation. But if we run our simulation, even if we have coupled everything, there's really nothing that displays that. Oh, sorry, this is important. I forgot. We need to enable the neighbor tracker in the XML for this simulation to run. So to enable the neighbor tracker, we need to come to the XML model, do CC3D ML, plugins, and neighbor tracker. That's it. That is an additional plugin that I forgot we had to initialize at the beginning when we were initializing the simulation. But again, if we forget to initialize a particular plugin that might be necessary later, we can always come to the XML and add it. So yeah, if we run our simulation now, the model is actually solving the simulation is actually solving the delta notch models, but we have no particular way of visualizing how it's been resolved. So to be able to visualize it, we will do what we did in the previous example, where we added a auxiliary field to be able to see it, to see the values of delta and notch, in this particular case of delta and notch in each of our cells. I'm gonna go out a little bit quick over this because we've done it already a couple of times. But just as a reminder, in the init function, we go to CC3D Python, extra fields automatic tracking, and we need 2 of those. Right? And then let's call one delta. And call the other one notch. And for the attributes, we'll just call them D and N. And then as Paulie pointed out in the previous example, always good to initialize the attributes in the start function. So we will initialize the attributes D and N for every one of our cells. And then when we iterate over our cells, what we will do is that we will update how to add cell tracking. So CC3D Python, extra field automatic tracking, and that is done in the init function. Right, and we need 2 of those, one for delta, one for notch. Then we've created these 2 attributes in the cell dictionary, D and N. And then now we have to update those attributes in the step function. I'm sorry about that. In the step function, when we iterate over our cells, then we're just gonna update the values of delta and notch by referencing the values inside each cell. And again, to reference those, we can always come to CC3D Python, SBML solvers, get SBML value, get SVML value for SVML in a specific cell, right? So we need that reference. And we call it D in the SVML model. And N in the SVML model. And I launch the poll just to make sure that everyone is with me. Oh yeah, I'm sorry. It's DN model. Good catch. It's not model name, it's DN model, right? Good catch.
Sorry, maybe you mentioned it before, but maybe I didn't get it. Here we use cell.spml. So does it related to the SPML markup? I mean, that language, or is it Antimony that we are using here?
So the reason it's called cell.sbml is also explained here in the comments. I didn't go into it and I'm sorry, but the reason it's because even though we declared the model as an antimony string, CompuCell translates it into an SBML model and then into an SBML model. And then we, after loading it, we can just reference it as an SBML model. So that's why we reference it using that SBML.
Okay, you mean that when we write it as an antimony, then automatically it converts it to the SPML, then
Yes.
we need to call that function because of
Right.
that. Okay, thank you.
Yep. And I'm actually giving time. The reason I'm paused is because I'm giving time for the people who are still working on it. So now, if we come back to, just to make sure, because I had to take off my AirPods, I ran out of battery. Everyone hears me okay?
Yeah.
Okay, good. So now if we rerun our simulation, there was a typo. Somewhere I typed dig instead of dig. Just to make sure that I didn't repeat the error anywhere else. Yeah, I did. It's just a typo on my part. So now I should be able to see delta and notch. From my previous simulation, in my previous simulation, we fixed the values of D and N or the variables that we were using. So I have to unfix them here now. If I want to be able to see the actual patterning. And again, to fix the color bars, I always do this in the preferences. And the last thing, so now if I run it, if I run it, nothing is going to happen because I've actually not initialized any values of delta and notch in our cells. So even though our cells have the capability of implementing the delta notch model, we haven't given them any delta or any notch. So what we will do to initialize them is that in the start function, there's two ways we can go about it. We can initialize them in the antimony string, in which case all of the cells are going to start with the same values. But what I really want to do is I want to initialize each cell with its own value of delta and notch. And what I'll do is that in the start function, when I'm iterating over my cells, I'm gonna sample from a uniform distribution. I have it here. To initialize each cell with a random number or state for the delta and notch variables. And note that to do that, I need to import an additional library that is not immediately available in CompuCell, and this is a Python library, and it's called Numpy. Probably some of you are already familiar with it. Probably some of you know it even better than I do. But yeah, what we will do is that we will import this Numpy library to be able to sample from this uniform distribution to start our delta and notch variables. So all I did is I imported the library Numpy as NP. And then in when I'm initializing my cells, I assign a random number, a random value between 0.9 and 1 to the notch and delta in each of the cells. And I've just realized that if I run this model, it's going to crash. And the reason it's going to crash is because I am referencing the SBML model before loading it to the cells. So I need to move the loading function before I actually assign the values, the initial values for each cell. Okay, so just to make sure, I'm gonna repeat what I did here. So up to here, we had initialized the SBML model in all of our cells, but we haven't initialized any values of delta and notch. What we're doing now is that we are initializing delta and notch to be a random number between 0.9 and 1 in each of our cells. And to do that, I'm using this Numpy library that I just imported. And assigning a random number between 0.9 and 1 to initialize those values. Okay, I'm waiting for, there's a couple of people still working on it. I'm just waiting on people to catch up. For those of you who have already caught up with me, I would recommend you to run the model yourself and then write on the chat, what do you see when you display delta and notch? So AJ, are you seeing some sort of pattern? Good. So I'm gonna run the simulation myself. And see what we get. So I do see some type of patterning, but it's kind of incomplete here. Let's launch it again. That's delta. And that's notch. So it looks more like this poly. That was just a matter of what the initial condition, random initial conditions were. But now you can see that that delta, that those cells that are high in delta, which are the red ones, are also low in notch. And the cells that are high in notch are also low in delta. And that results in a patterning. If you have a little bit of a different, I would recommend that you go back to the equation. When you wrote the delta, the antimony string, just make sure that the antimony string resembles this one, which matches the equations, and that you're calculating the average delta of the neighbors correctly. Okay, good. Okay, how can we say this as a video? Excellent question. I don't know how this works in NanoHub, but in a local version, you would come to your configuration settings, output, and here you have the option to save the images every number of Monte Carlo steps. So at least locally, when I do it, this stores images of at the frequency that I specify here, saves PNGs of the simulation domain as it's being as the simulation goes by. And then once I have all of those images, then we usually take all of those images and use external software to turn them into movies. Okay, again, if it was a bit of a complicated exercise, so if your simulation does not matches these results, there's I would recommend you to go back to the code and make sure that there are no typos in your model string, no typos in when you're calculating your delta average. And there is a I've uploaded exercise 413 to as an exercise so you can reference it back to make sure that you have no typos. So the last thing I want to show, and I'm just going to show really quick, is how we can have access to some of these models that we were looking at. There is a so okay, the second part of this exercise is to add steering panels. I'm not exactly sure. I'm actually not sure what I should do next. So Tarim, please make sure that your visualization settings are correct. So make sure that when you come to your CC3D preferences and you come to your fields, that none of the values are fixed. If they are fixed, you might not actually be able to see the fluctuation, the changes in delta notch. So that's a third reason you might not be seeing the patterning. So if the code is the same, it's working, make sure that your visualization are is working, and this is what you should expect to see. So in the next exercise, this is not complicated because it builds on this one. I will show how to add steering panels that allows us to modify the parameters of the delta notch on the fly. And on the fly, I mean as the simulation is running. And the reason we might want to do that is because, as James explained earlier, when you build these models, you might build them in explanatory, at an explanatory stage. And in that explanatory stage, you might want to explore how changes in the parameter values affect your simulation. So that's what we're gonna do now. So just keep if your simulation is working, we'll continue working on that one. If not, you can always start working from the demo exercise 413, and then we can work together from that one to put the steering panels, to add the steering panels. Oh, James had a good point. Once you've, for those of you interested in saving images, you have to click on save image every nth Monte Carlo steps, and you specify the frequency you want to save them. And once you do that, you hit on the picture on the camera icon in all of the windows that you want to save. And then, at least in my local version, every time I run CompuCell, that creates a directory in the CC3D workspace corresponding to that simulation. And then the pictures of the simulation are saved into that folder. Okay, so coming back to adding steering panels, the first thing we need to do to add a steering panel is to define this add steering panel function inside inside the step away. So what we will do is that we will define the add steering panel function, and that's below all of the other functions in our step-away class. It takes self as an input. And then we need to specify which of the parameters, or what are the names and values and ranges of the parameters that we're gonna include in the steering panel. And the way we do that is using this add steering param function that takes the name of the parameter, and we're gonna call it A, because delta notch has a parameter called A. The value is the reference value that the simulation is gonna start with, or the steering panel is gonna start with. We're gonna set that one to 0.1. The minimum value of we're gonna let this parameter change. It's gonna be called min val. Gonna set that one to 0. And the maximum value, we're gonna let that parameter change. Max val, we're gonna set that one to 0.02. The decimal precision of the steering, in this particular case, is gonna be 3 decimal spaces. And then the name of the widget type that we wanna use. And that we have different widgets. We have pull down menus, we have sliders. In this case, we're gonna use a slider. So this creates a steering panel for parameter A. And I'll use this as a template to create another steering panel for parameter B. So again, I changed the name of the parameter B, and in this case, it's gonna go the default value is 100, the minimum value is 0, the maximum value is 200, and the decimal precision is 0, which means that the increases are integers from 0 to 200, and also a slider. Now, if I rerun my simulation, I should be able to get a steering panel that allows me to change the values of parameter A and parameter B. And just notice that parameter B, the maximum value is 100, and I wanted it to be 200. I'm gonna launch the poll to make sure that people are with me. All I did was I added this code to my previous simulation to be able to generate the steering panels. So if you can notice that even though I created the steering panels are not changed as I change the values of the steering panels. the steering panels are not really changing anything in my simulation. And this is because even though I created the panel, I haven't specified how the changes in this panel changes any value of my simulation. So that's what I need to do next. Next, I need to specify what the value of in the status panel corresponds to in my simulation. To do that, I need to add a second function called process steering panel data, take self as an input. And then to get the value, what value I'm passing from the steering panel, I need this function, self get steering param, which basically is reading the value that I'm passing from the panel in A. And in B. And I'm gonna store those values in these variables A and B. And now that I have the value, all I need to do is to update the values of the parameter inside the SPML models in my cells. I have multiple options. I could create whatever conditions I want, and I could choose whatever cells I want to change that parameter in, but I'm gonna just change it in, I'm gonna change it in all of my cells at the same time. What cells you change your parameters in, of course, are gonna depend on the particular program of interest. Here, just for simplicity, I'm just gonna change it in all of my cells. And this is just the same syntax that we use to update the D bar, but in this case, we're gonna update A and we're gonna update B. Okay, I'm gonna relaunch the poll. So now if I run my simulation, gonna let it settle until my cells are either high in notch or high in delta. And now I can start exploring how changing the parameters of might change the values in my simulation. It's not doing anything well. Let's see. Okay, let's make sure that I'm doing it correctly. I do have the parameters A and B. Yeah. Not exactly sure what's happening because it was working earlier, but I have a working version that shows how this is supposed to look like is exercise 414. And again, if I run my simulation delta and let it settle. Now we can change the parameters. Let's see how that changes. Our model. So if I set A to 0, I lose my patterning. And then the more I lower beta, the higher everyone is in delta. This is of course related to what A and beta correspond to in the model, in the OD model that I showed earlier. But I can probably, by exploring this, I am basically exploring how the magnitude of the activation and inhibition of delta and notch, what the effect of changing the magnitude of the inhibition and activation are in the pattern formation. That allows me to explore certain biological hypotheses about, okay, if this is my the steady state of my system, what happens if I perturb it in this direction, if I increase the inhibition of notch on delta, or if I increase the activation of delta and notch. Oh, I'm sorry. Okay. So I'm gonna stop this simulation, this exercise here. Again, everyone for people who want to try it at home, it's exercise 414. So the last thing I wanted to show in this module was how we could look up for some of these SBML models. We've shown previously in the previous simulation, in the last couple of days, we showed how to make our cells divide in CompuCell, and we set a very simple condition for our cells to divide. We asked the question, have our cells double their size? And if they had, then we will trigger cell division. But in reality, there are multiple checkpoints that cell has to go through, and to be able to go through the full cell cycle and divide. And there are markers that indicate that the cell has successfully gone through some of these checkpoints. And the cycling The proteins called cyclins are regulators of the cell cycle, and they're also markers that the cell has gone from one phase to another in their cell cycle. There are mathematical models of the cell cycle that we could use to incorporate into CompuCell, and then make for example, if we wanted to explore more complicated models of cell division, we could potentially include the cell cycle models into our cells and then base the conditions of the cell division on those internal cell cycle models. And there is this particular model that was published by Tyson and Novak in 2001 that has a bunch of differential equations. But from those two are really important because they show the oscillations that we would need to characterize the cell cycle. The details are complicated, and we're not gonna look into the details of that particular model. But what is important, what I did wanted to show is how we can have access and how we can look up for this model. And there is a repository of models called biomodels, where we can go to and we can look up for SPML models of interest if we wanted to look for more intracellular or subcellular models. We just type the type of model that we're looking at, or if we know what the identifier of the model is, we could also look for it. In this particular case, we were looking for the cell cycle regulation model, which has this identifier. Once we have identified the model, we can go into the model and the files and look for the XML file that contains the model as an SBML. And then what we do, once we have that SBML, we can import it into our CompuCell simulation and load it into our cells. Let's say we have a single cell that we want to load this SBML model into. All we need to do is we have this functionality, add SBMLs to cell, that allow us to load the SBML model directly, even if we didn't specify it as an antimony string. Let's say we have an SBML model, our SBML file that has the model. Then we can load it directly as an SBML by referencing the model file. So far, we have used a model string, but in this particular example, since we have it as an additional file, we can load it into our cells. Then we can display whatever variables of that model we want using the auxiliary fields. And if we load it in a single cell and run it, what we will see is that the values of the cyclin B are gonna just increase and decrease continuously. So it's gonna oscillate from going up and down. And since we know that the cyclin B is going up and down, then we can establish a condition for cell division. So for example, in this condition, we say that if cyclin B has dropped below a threshold, the 0.5 threshold, and this is where we have established the condition. Remember, we do this in the mitosis step already. Then we trigger cell division on that cell. So basically, we have put the cell cycle model inside each cell. We're measuring the value of the cyclin B. If cyclin B drops below 0.5, then we divide that cell. And if we execute that simulation, what we will see is that the cyclin B starts increasing, and once it decreases, the cell divides. And then now we have two cells with the same amount of cyclin B, And when the cyclone B in that cells increase, when the cyclone B decreases, then those cells divide as well, and so on until they fill the whole simulation. And this example is available for you in exercise 416. Of course, there's interesting questions that you could ask about this model. What happens, how we could implement different values of cyclone B in each cell, because we know that the cells do not go, even cells that are seated at the same time do not go over the cell cycle all at the same time. So this is an approximation, but we could, those kind of things you could potentially do, but by rewriting the values of the cyclone B inside each model, inside each cell. Okay, and that's the end of my simulation, and I'm gonna thank you for listening and for sticking with me, and I'm gonna hand back to James now. So there is a question, and I'm pretty sure TJ, and I actually have that question too. Yeah, thank you for asking it. Does CompuCell support integrating the SBMLs with a stochastic simulator?
Yeah, this is a great question and a very new feature of CompuCell. So the simulator that we use, which is the same as what's used in Tellurium, actually has a whole array of integrators that are available that you can select as inputs. And one of those is a Gillespie solver. So if I remember correctly, there are 5 available in libRoadrunner, which is the simulator, and 4 of them are deterministic, and 1 is the Gillespie algorithm. And the way that you can select that is that in each of these functions that are available for creating antimony or SBML models and attaching them to cells, each of them, they all have an optional keyword argument called integrator. Remember correctly? And that keyword argument integrator, you can pass to that the names of the different solvers that are available, and one of them is Gillespie. So I'll type this into the chat. You can enter that. And I'm frantically searching through our manual to find you the reference on that, just for your own reference. And but of course I can't find it off the top of my head. So yeah, if you want to shoot me an email and I'll send you a link. But yeah, that's available for antimony, SBML, and CellML models. And then in one of the modules that we'll cover tomorrow, that's also available for our Boolean network model simulator, which is has Mabos is the backend on that. That also uses a Gillespie integrator. Tarun asks, is the integrator available in CC3D Python? Yes. So when you type, say, cell.sbml and then give the name of a particular model, that actually gives you the entire solver, which has the integrator attached to it. So you can feasibly write cell.sbml dot whatever the name of your model is, and then dot integrator, and that is the actual integrator that's being used.
Hi there, I have a question.
Please.
So I wanted to go back to the thing about in the notch delta model about iterating over the neighbors and the thing where it said common surface area. What
I'll
I
turn
wanted
it
to,
back to Josh then. Okay,
Yeah.
Josh, you want a screen share?
Yeah, 1 second. let me get that part here.
Yeah. So I thought that's what I didn't understand is whether, in fact, as it's been written, the program, it is actually using that feature, or some additional code would have had to be put in to take advantage of that feature. Because it seems, from a physiological point of view, that is a good feature, because if two cells are in contact with each other, it's only the delta that is on the surface of the adjacent cell, which is in contact, that is relevant to the amount of notch signaling that a cell receives. So rather than counting all the delta that is in the neighboring cell, just what's in the contact area should be important. So is that what the way it's written, it's doing it, or does something additional need to be written for that to happen? I can't.
So yeah, so when the for loop, it's iterating over right, the get cell neighbor data list gives you that information, so it's already available to you. Whether you decide to use it, as I mentioned, it's up to the user, right? So for example, we could have divided where we have df there,
Yeah.
we could have divided by that variable common surface area. Because that would make sense, right? And that's what I was explaining earlier. It would make sense that if that the how much of the delta it's contributing to the signal depends on what the common surface area is between the cells, because these are
Correct.
cell
Correct.
surface receptor, right?
Correct. Correct.
So it would make sense to divide the common surface area by that, at least in this particular example. But it's available to you because the for loop is already iterating both over the neighbor, the reference of the neighbor, and what is the common surface area between your cell and the neighbor. So yeah, it's available to you.
Okay, thanks.
Mhm.